All right, so we've really only talked about like half of immunity at this point. And you might have noticed that we're on part 6 of 7, and the part 7 is sort of adding up immunity. So we are not getting into actually the favorite part of immunity that I've done, which is T-cell MHC interactions. But I want you to at least know what T cells are, what MHC is, especially because I did my graduate work on it, and my postdoc was actually on a related, it's an MHC-like molecule. Now I had to cut all that stuff out this year, but I do want you still to know about this other system, and it's really important because one of the things I was saying to people over and over during COVID was we get all this data about antibodies, and there's all these things about what antibody levels and stuff like that, and that really is important because the antibodies are fast-acting, they're protein-based, you make a lot of them, they're really good at what they do. But that's not all of what the immune system is. In fact, my whole study was on a branch of immune system. I never used antibodies until like 3 years ago. Well, technically, I did use them in small extensive things, but I never had a focused study on antibodies. Now, we're doing that this year. We're doing antibodies this year in the lab. So I want you to have more of it, and I'm happy to do more of it. But I want you to say that I was decades in structural immunology, not focused on antibodies at all. And I want to give credit to my advisor who passed away a long time ago now, 2019. I think I found out during COVID because I remember finding out like early on in COVID, but you see that it was definitely before COVID. And he was actually my advisor at University of Washington, and he really focused on mentoring. And one of the reasons why I was able to figure out what's going on with immunology is because he invested in me first. And he had a really great lab environment where I could learn this stuff. Because immunology is never really taught at the undergraduate level. I mean, it's a lot of what we've done to have 3 days devoted to structural immunology, but we're really only scratching the surface. And it's so important, I want you all to know, and this is the kind of thing that I think will help you. There's a lot of activity in this area. I'm going to tell you at the end of this about a job that a student who was sitting in this class 20 years ago, a job that this student is doing now, based on his undergraduate degree, doing one of the most cutting-edge T-cell therapies. Just to ask you before we all get started, have you heard of T-cell therapies as a kind of thing? So do you know someone personally? Or
yeah, what is it? He actually went to SVU.
Yeah.
He's fighting AML leukemia right now, so he's at that stage where they're looking for a lot of experimental stuff. So they're looking at the T-cell treatment, and they're also looking at something else, and I can't remember what he was talking about.
Okay.
I think it was mentioned in lecture, but I don't know which one.
Yeah, perfect. So immunotherapy is really great for that kind of thing. So hopefully it will, and if you know more about it, that might even help you make choices down the road. So it's that kind of thing. Biochemistry actually has big relevance here. Have either of you heard of T-cell based therapies? So, okay, so that's good. We'll talk about them a little more, and I'll have a little bit about it, but it's something that makes people money. So based on just this class, this actually can't prepare you enough. Now, I do want to connect it to the antibody. They went through a lot of trouble in Wells and Company. They went through a lot of trouble to find the CDCP protein that was a difference on the surface so they could use antibodies against it, but the difference on the surface reflected a bigger difference inside the cell because it was a signaling difference. It wasn't necessarily outside the cell. And so it took them a lot, and they did all that because they wanted to use antibodies. Well, there actually is a system that your body has for the immune system to look inside the cell. And the thing is, this is not mediated by proteins, but it's mediated by cellular interactions. And so you have to take care of cells. If you're doing a T-cell therapy, you have to extract cells, modify them, put them back in. So T-cell therapy is automatically younger than like Genentech got its start doing antibody stuff. Antibodies are automatically easier. The words for this is humoral immunity because the humoral sort of things, the antibodies can be humoral is the word that they remember that they're bodies because they were not cells way back when they got their name. But this branch of immunity is cellular immunity because it requires an entire cell to work. But it can do some really cool stuff. Also, you can imagine it's more complex, so it takes a longer time. And that's why you heard all these studies about antibodies in COVID. But it's harder and it takes longer for T cells to kick in. The good news is now, a couple years after COVID, hopefully our T cells are educated as a population. And I think that's some evidence for the fact that we seem to have built a successful wall against the virus coming back. Let's hope. And there's reasons to think that this wall will be a strong one because of the way T cells work. So just remember immune response, and we aren't even talking about all the immune response. There's like at least 2 other areas of immune response that we could talk about. Oh yeah, there's at least 4 or 5 other areas. The T cells and B cells are the big area. There's different kinds of T cells. But I studied as a postdoc natural killer cells, which is a third kind of area. Both B cells and T cells are adaptive immunity, so they're like the smartest part of the system. They can actually adapt to something they've never seen before. So when you see what the immune system's job description is, it's a pretty big job description. Basically, keep the body intact, attack anything that's not the body, except if it's like a good E. coli, then you don't want to attack it. You need to make these decisions about what to attack and what not to. And as anybody knows, making those decisions is one of the hardest parts of any kind of strategy. So an infectious agent, you want to react against it and have protective immunity. If you have a deficient response, you have a recurrent infection. You remember all the worries about recurrent infections from COVID. Thankfully, it was still a pretty big deal for some people, but it wasn't as big as the worried voices talked about. An innocuous substance, you want there to be no response. Basically, you want it to be on in this case and off in that case. If you do have a response, you have a problem, shown in red, an allergy. Hopefully, the pollen is treating you all well. I know it's just all burst out this week, but it's a protein, and your immune system says, I've got so much of this pollen in my system, there must be something wrong with it. And sometimes people freak out against that. The systems do. If you have an organ transplant, well, there you actually want to turn off the immune system because you want the person to accept it, and that's why you want to get someone whose immune system matches if you're going to be doing that. And of course, for your own organs, you want to be able to have your immune system not attacking them. But sometimes it accidentally starts to attack. And then you have autoimmunity when you're attacking yourself. And then on the other hand, if you have a tumor, the tumor is supposed to give you immunity. You want your body to attack the tumor. But the tumor is basically your own cell gone bad. So how is the body supposed to do that? Remember that there's not many differences on the surface of the cell, but there are some. Wells found one, and your body finds some sometimes. Who knows how many tumors we have that start to get a foothold, but then our immune system actually is able to basically eliminate them before we even know that there was one that was there. So, and when I first started, I was like, why are they studying an immune system at a cancer center? And I realized later it's one of the most important things to study. So, the immune system has to strike a balance between reacting to foreign agents and not reacting to yourself. And it's really interesting because if you think about the philosophy, a big point in philosophy is what is the self? Who am I? My identity and things like that. Well, in a sense, the immune system has a biochemical version of that philosophical question. Who am I? And as you're growing, and even in the process of making a new human being, the immune cell actually plays a role in the development of a baby, and that's mammalian species. Natural killer cells actually play an important role in forming the placenta. So there's all sorts of things that it does, but if you think about it, there always is a balance. We have to interact with the outside world. If we don't interact enough, we get infected by it. If we interact too much, we have inflammation, which causes tissue damage. So it's really kind of a balance between damaging the tissue and preventing abnormal growth, whether it's the growth of a virus or the growth of my own cell that's been turned on to grow too much. the immune system can actually do all these things, and it's kind of a really interesting place philosophically. I encourage you to think about this with all your liberal arts as you think about it, because it's a fascinating system. It's amazing to even think how it evolved. And the balances that you can talk about in the immune system can take place in the immune system signaling molecules. We don't spend much time on interleukins, but you do have different types of responses that involve different types of interleukins, making different types of cells be activated. And so you want this balance to be, this is why these signaling molecules are important. There was an interleukin called interleukin 6. In severe cases of COVID, it would go haywire, and it would cause the whole immune system to attack yourself. In that case, it's not so much, the virus is the trigger, but the real knife edge is your own cells overreacting and producing a cytokine storm. This has gotten out of balance. And so you just want to think about all these cases that it has to go to. Immunology is actually a really fascinating area to study. If you're thinking about what do you study or what do you go to work in, I do want to say that some of these lectures might be some of the most useful. At least I'm hoping they are useful to you someday. Okay, but we're not going to talk about details like that. Let's talk about medical details, though. Okay, so the thymus is an immune organ. We have not talked about it much yet. Does anybody know where your thymus is? Anybody want to point to it? Down a little lower, but it's in the center. It's basically above your heart. It's kind of where some people think their heart is. And I guess that's the heart of your immune response. Do you know what happens to you if you remove it? At least I have what happens to mice if you remove it. Any ideas? I have a picture of it, right, by the way. I mean, let's make a prediction of what would happen just in very general terms. What do you think?
More prone to infection or inflammation or things like that because you don't have the regulator anymore?
Yeah, so infection would be one of the, and the thing is, if it's a deficiency, you have infection. If you have an overreaction, you have inflammation. But you actually can have both. If you're just removing the thymus, you might think infection is the first thing that happens, but inflammation can also happen. Savannah?
Would your lymph nodes get inflamed because they're being overused?
Yeah, when your lymph nodes swell. Now, I don't think your thymus itself, because your thymus is on the inside of your body, I'm not really sure if it ever actually swells like a lymph node, but it seems to me like there's got to be at least some disorder where it does that. And yeah, the lymph nodes are connected to the thymus, so everything you know about lymph, do think about how it's connected to that. And actually, my wife had cancer, and they were worried about spreading, and they checked the lymph nodes, and they actually found a couple that had a little bit of cancer in them, and they took them all out. I think it's amazing that we can do that. So, yeah. Other thoughts or ideas about the thymus, like what would happen if you had a thymectomy?
Would it affect T cell production?
Would it
affect T cell production?
Oh yeah. It would totally. The thing is, you would still have T cells. They just wouldn't have school. It's like you're taking away the T cell school because they're produced in the blood marrow, right? But yeah, it would affect how they work, and probably they would be sick and they wouldn't survive as long. So it definitely would. You'd probably have fewer T cells. So let's see what happens. And honestly, this is what happens with mice. But with mice, it basically looks like a thyme plant. I don't know. I guess that kind of looks like a thyme plant, but that's where it got its name from. It's got its name from thyme itself. And so is the gland, and they basically took it apart. And when you take it out from neonatal mice, just right after birth, basically the white blood cells drop dramatically, so exactly what you were saying. And the animals, they became susceptible to infections, exactly like the rest of us were saying. B cells dropped in number, but there was another white cell they still had some B cells. There's another white cell that basically completely disappeared. Then they died of mouse hepatitis. Many had bacterial pathogens in their spleens. And they realized that the different type of cell was actually more connected to the thymus than B cells themselves. And so those are T cells. And this is a picture. My son found some kind of anime that was about the immune system, like personified. And someday I've got to watch that and like critique it scientifically. But The thing is, you have the B cells. Here's my natural killer cells, and you have 3 kinds of T cells listed. They have a superhero team of lymphocytes, and 3 of them are T cells. That means that the lymphocytes can play different roles. And look how you have the CD8 cytotoxic T cell that looks kind of like the natural killer cell. Cytotoxic, if you take that apart, also a natural killing function. But then you have the other ones on this side that are more like regulatory cells. And in fact, this would be one of those superheroes that doesn't really use a weapon. Maybe it's like Batman not using a gun. They're helper cells that more are about coordinating other people and communicating than they are about actually killing cytotoxic functions. And so the important thing, this is something that I kept telling people during COVID, that you have multiple issues of your immune system. And this doesn't even show antibodies in it. Antibodies are early in this thing as well. When you get infected with the virus, the first thing that happens is you have sort of natural triggers that are set up. The virus is causing damage, and you have sort of automatic response systems. Those are all these signaling molecules, and NK cells are sort of fast responders as well. If you already have antibodies, then you will make them at this point. If you don't, it'll take you a little bit longer. And the virus is basically going through your system. These initial responses keep your virus from completely overloading things, but then it establishes a foothold. And if they can't take care of it, you need to have something that adapts to the virus to kick it out. And that's what T cells can do. After a week, the T cells start to go up, and as they go up, the virus goes down. And this is the time when you have like a bad cold, or the first time you had a cold post-COVID, things like that. Probably it was a 7-day cold, because probably you needed to sort of have your T cells wake up a little more. So different cells have different speeds, basically. And you have the adaptive response takes 7 to 10 days, but it also remembers. Years later, it will be faster, much faster. You will have it responding in a couple days if you have already seen that virus. When COVID started, I'm like, okay, well, here's all my stuff about immunology is going to be put to the test, because I'm going to be able to say your immune system should be able to handle this eventually. It's a very nasty, very transmissible virus. But there was a lot of people that were saying it would be like a world ender. It would be something like we've never seen before. But I knew we had seen it before, at least 60% of it before. So no matter how bad it was, I knew that the immune system would eventually come and put up a fight. I just was worried about how bad it would be before that happened. It was pretty bad, right? So, but we're in a different place now, and I think the immune system has really helped us. So these are the different types. This is actually from your Leninger textbook. And so I just wanted to mention there's a little bit about immunity in there. And this lists B cells and T cells, the 2 types of T cells. It also lists macrophages. Remember we talked about those. Those are one of the first kinds to be discovered, the phagocytic cells that ingest large particles. They just basically phagocytose it, they eat it up, and they chew it up, and they spit it out, and then they help the other cells. So they crawl around and they collect stuff. Just notice that technically you don't really have that many major types of B cells, but you have a ton of different types of T cells. One of the other reasons why T cell therapy has taken a longer time than antibody therapy. Here's some examples of it coming all the way from the blood. Look on the right here, and you see red blood cells. And on the left, you see the same cell can actually make white blood cells on the left and red blood cells on the right. When you look down here, you see the B cells and the T cells, and they both come from the same progenitor. They're basically cousins, although they have very different functions. You will see a lot of similarities between their receptors. The T cell receptor, you might predict, would look something like an antibody, and you would be completely right. So you have the issue of this. I want you to see the red blood cells are actually sort of a slimmed down version of a white blood cell with a lot of hemoglobin in them. That's their only function, basically. Macrophages are down here. They come from a different branch of the family tree. And they look like this. I love this textbook. By the way, I've had students who started a job where they need to learn immunity, and 3 lectures in biochemistry doesn't really cut it for them. And I do have a textbook that I really like. If you ever want, you can even borrow mine. There's a really good textbook because it has very clear pictures, but these are also realistic pictures. These are what the cells look like. And I think that really helps rather than to have just like a sphere. But for other types of learning, you can have more slimmed down pictures. This is just another one, and I just like what this one looks like. And just see that the lymphocytes are the class that includes B cells, T cells, and natural killer cells. We've got a whole bunch of immunity that honestly I don't know that much about. The one thing that you want to know about is down here, macrophages, which we've mentioned, and dendritic cells, which are like macrophage cousins. But they are the ones that actually connect back over here. All the other stuff, don't worry about it, because I don't even really know. We'll mention a few things like mast cells and stuff like that, but it's not the kind of thing that we focus on. But when you talk about T cells, the question is, all this stuff, could I give this lecture to you before, in Biochem 1, based on everything that you learned from that? And really, most of it I probably could, but there's parts of it that have to do with Biochem 2. Because we're through Biochem 2, at least partially, I would like to mention what those parts are. So T cells, remember that they get educated. They start off naive, and then they figure out that when they're useful against a particular infection, they proliferate. They become effector T cells that are fighting off the virus, like in the previous slide. But then over time, the virus goes away, you don't need as many T cells anymore, and so they switch to memory T cells, and only a few of them hang around, but they're waiting just in case that virus ever comes back. Each of these different kinds of cells has a different kind of metabolism. Kind of like your metabolism changes as you age. I regret to tell you that the metabolism that you have right now is probably going to slow down in some ways. It's going to become more anabolic, about building rather than catabolic. So the thing is, naive T cells, they basically are under catabolic metabolism. It's sort of the hovering thing. When they need to grow, they become anabolic metabolism. And then when they switch to memory, they don't need to grow anymore. They just need to hang around. So again, they switch back to catabolic. And you can even get into exactly what parts of the system are. So I want to show you this just because you might think that the Krebs cycle doesn't have much to do with immunology, but it actually does. The cycle is right here, and hopefully after Biochem 2, my goal for the class is for this to sort of automatically make sense to you. You don't have to understand everything, but at least you can see, oh, glycolysis here, Krebs cycle here, oh, there's pyruvate and oxphos here. You can sort of put it together and understand that if you have a cell, like this is a normal situation for a cell, and you have different things that the cell feeds on and puts out. That's the whole point of Biochem 2, is to be able to take something like this apart. When you're under aerobic glycolysis, you actually are taking in a lot of pyruvate and taking in glutamine. So even that chapter of Biochem 2 matters. You're putting out a little bit of acetyl-CoA, and it's making lipids and stuff like that. This is how an immune system cell changes when you go from glycolysis to aerobic glycolysis, which uses oxygen. And so when you have all these different kinds of cells have different kinds of metabolism on the inside. And so I think it's kind of cool. I mean, the naive ones just have a simple metabolism. The effector lymphocytes get hungry for more glutamine, and they put out more lactate. And they make fatty acids inside them. They're growing. Of course they're making all these components. It makes sense. Memory T cells are back to the original, but they make a lot of glycerol because they want to be around for the long haul. You can come up with reasons for any of this. And by the way, we are going to do the Biochem exit exam. Maybe I could do an exercise that's related to this, because I want you to be able to bring back these things and say, hey, why would a macrophage be making fatty acids? That's interesting. Or why would a neutrophil be putting out lactate? You know, it's that kind of thing. But just realizing each of these is different. And then you even have different types of metabolism for different types of T cells. Here's the spectrum of T cells that you can get. These are T effector cells, which notice they all have a sub H. These are all different kinds of helpers. And then there are antigen experienced T cells, Tregs are an important class, and I think none of you knows Nick. What's his last name? There's a student whose first name was Nick, but he graduated in like 2010. But he just recently, like a year ago, got his PhD from the Hutch, and he went to Pelton Auditorium. I forget his last name, but I'll look it up. It was Nick. So if you ever meet a guy about 10 years older than you, and he was in this class as well, he was a research student, he even used Bucor. But you have the, he studied Tregs, I believe. So we have all these things that people can focus on and learn about. But the important thing is that these have different types of metabolism. These guys like to use glycolysis. These guys like to use oxphos and fatty acid oxidation. These ones also like to do all three. So, but those are alloreactive, which usually you want to turn those guys down. Maybe that can be a way we can look at that. So all that's just a touch onto the idea, and maybe, if you have metabolism topics, I do encourage you to lean into that for this class, because that's a good way to bring everything together. So when you look at the receptors, though, remember how I said B cells and T cells are cousins? And B cells, of course, have an antibody-like receptor on their surface. T cells have a much smaller receptor on their surface, but it looks just like one antibody arm. In fact, it has the same IG domains in it, and it has the same antigen binding site on the end. And so you would predict from this, how many loops do you think it has? 3 on one, 3 on the other, you have 6 total. So yeah, the rest of it's connected directly to the membrane, but there's something very similar about T cell receptors. But of course they work very differently because they don't make soluble antibodies, they recognize an antigen on another cell. So like when a cell is infected with a virus, the virus is hiding out inside the cell. It might cause a cell surface protein to be dysregulated, but if it did, and the immune system found it out, the virus would evolve a way not to do that anymore. So there's a system, though, that takes the inside of the cell and it puts it on the surface. Like this virus is infecting the cell, and it's going out and making other viruses. But the cytotoxic T cell recognizes these little red things. And these little red things are bits of the virus that have ended up on the surface of the cell. And the virus doesn't want that to happen. Your body makes that happen. And so what's going on here is that you have this system. What is this system that actually puts stuff on the surface of the cell? And you can actually see, here are viruses budding out of infected cells. Here's a virus-infected cell with a T cell clearly recognizing something on the surface. If the surface is that flat and they're that close, you know that something's going on. And so what's going on from the molecular level is what we want to talk about. This can even be used for cancer. They talked about viruses, and if you have a cancer cell that causes, if this is a system that's monitoring the entire inside of the cell, if a cancer causes a problem inside the cell, then that could also be put on this system to be displayed on the surface of the cell. So like here's and in fact we could possibly use this for a cancer vaccine. It seems like a crazy idea, but it actually makes sense. If the immune system can respond against cancer, we can train it to respond against cancer just like we train it to respond against viruses. It's just a question of putting the part that gets on the surface of the cell into the cell in the first place. And then that part gets displayed, and the T cells say, hey, this is bad stuff. And then they get activated, and they go over here to a cancer cell, and they see the same yellow thing. And then they're like, oh, well, we're going to activate, and that's where the cytotoxicity happens after that. So what is this system that takes stuff from inside the cell, and basically it's a system that samples all the proteins made by the cell. So normal proteins are sampled by this as well. The virus can't get away from it very easily. When a virus infects a cell, the one thing it has to do is replicate. When it replicates, it makes more viral proteins inside the cell. And so you have all these little red dots around. Now some of them become functional virus and they bud out and stuff like that. But not all of them are functional. Some of them are left over. And you take the leftover bits, you even chop them up a little bit to make them more manageable. And then you have proteins that bind the chopped up little bits. This yellow protein right here is the MHC protein. it will bind the red viral peptides, and then your cell will move it from the inside of the cell to the outside of the cell. Technically, it's in the ER if you care about cell anatomy that way. But then you have the little viral bit and a little yellow bit that is your own protein. Only the red part is really a foreign protein at this point. And so that means that the T cells have to recognize both of these. It's how the T cells know what to look at. The T cells bind both the MHC class I and the peptide. Remember, there's a lot of your own peptides that are caught up in this system and they're moving up. So there's a lot of your own peptides that are on here, and your T cells know not to freak out when they see one of those. That's what the thymus is for. The thymus is for them to learn what a normal antigen presentation is, what a normal cell looks like, and what a normal MHC looks like, and they learn to bind that. When they see something where they can bind to the MHC, but they don't recognize the peptide, then that is what triggers them to go off and to freak out. This is why the analogy, you notice that the one of them was dressed up like a policeman. This is a policeman that has the power to destroy the other cell. So it's that kind of thing. And in fact, I will take a slight break right here, because this is the time where it's time